ER and synthesis of exportable proteins. Although the process of protein synthesis takes place in cytosol, as you all know, through the interaction of polysomes, the mRNA and the tRNA, which carry the various amino acids. Polypeptide chain grows through a groove or tunnel in the large subunit that is linked directly to a channel in the ER membrane. Some stage of elongation, the polypeptide chain begin to emerge and is finally deposited into lumen of the ER. In this figure shown experiment using proteolytic enzymes on free and membrane bound ribosome. This experiment demonstrated the presence of both free and membrane-bound ribosome of a protected polypeptide chain of about 40 amino acids at the COOH end of the protein. Furthermore, in bound ribosome, the rest of the chain with the amino end was found to be protected inside the cysternal space where the protein is segregated. On the basis of these findings, it was thought that the polypeptide chain moves through the large ribosomal subunits, become attached to ER membrane, and then through a channel spanning the membrane enters the ER lumen. The signal for secretory protein resides in the secretory proteins resides in the nascent polypeptide, the signal theory. Because free and membrane-bound ribosomes are continuously interchanged and show no difference between them, what determines whether or not a ribosome binds to the ER? The present view, named the signal theory, postulates that the information resides in the end terminus of polypeptide chain. According to Blobel and Sabatini, the signal should be present in the messenger RNA at its 5' end. At this end, there should be a set of signal codons localized immediately after the initiation codon AUG. And it is only when the special signal peptide emerges that the large ribosomal unit of the ribosome become attached to ER membrane. The signal peptide is thought to recognize and interact with special receptor protein in the membrane, which form a tunnel through which the polypeptide penetrates into the lumen. The hydrophobic signal peptide is removed by a signal peptidase. An important clue the signal theory was provided by Milstein and colleagues in 1972. They conducted an experiment in which the synthesis of kappa light chain of immunoglobulin from mouse myeloma cells was followed in vitro on two different systems: reticulocytes containing no ER, ascitic cells lysate containing microsome. The analysis of synthesized protein showed that in system 1 the polypeptide was about 15 amino acid longer at the N-terminal end, wherein in system 2 the protein was shorter and of same size as the mature protein secreted by the myeloma cell. They concluded that A. The N-terminal sequence as a signal for the ribosomal attachment to ER, and this extra sequence is removed by an enzyme in the ER. Signal peptide is separated by a processing protease generally called the signal peptidase. This protease is associated with the membrane of ER and its active site faces the interior of this organelle. The signal peptidase processes the precursor polypeptide while it is being synthesized and translocated. This protease can solubilize by the detergent and purify by various chromatographic procedure. It is not specific and even a signal peptide from bacteria can act on eukaryotic cell. Now, The processed polypeptide continues to grow until it is completely segregated into the lumen. Some of the major molecular events that occur in the synthesis and translocation of polypeptide in the ER lumen. This diagram showing these events includes the translation of signal peptide, the association of ribosomes to the ER membrane, and the recognition of signal peptide by a possible receptor which act as translocator. The signal theory is supported by abundant evidence. Messenger RNAs for several secreted proteins have been translated on free ribosome and shown to produce larger polypeptide chains. These have been called pre-proteins to differentiate them from pro-proteins such as proinsulin, proalbumin, proparathyroid hormone and other, which are larger precursor of the secreted proteins. A mRNA for albumin first produces a pre-proalbumin. This, after removal of signal peptide, is converted into proalbumin and later on it again cleaved at a specific secretory stage to produce the final product and albumin. The signal peptides from different proteins differ in their amino acid sequence. The signal theory also applies to secretory proteins produced by plant cells such as gene from corn, glycinin from soybean and chymotrypsin inhibitor from tomato leaves and the seed reserve proteins from the common bean. Signal receptor protein, signal recognition particle. It has become evident that hydrophobic signal peptide does not possess the needed information to ensure its transfer through the membrane. This implies that some other factor should be present in the system. This was first suggested by experiment in which microsomes were either extracted with high salt solution or were mildly treated with proteolytic enzymes. In both cases, the translocation was prevented and it was restored by adding back the extract. The factor removed by this treatment is a large complex of proteins and RNA of about 250,000 daltons, which acts on the translocation. This complex, which consists of 6 polypeptides and contains one RNA molecule with 114 nucleotides belonging to 7S cytoplasmic RNA type, it is so-called signal recognition particle SRP. The SRP aids in decoding of information carried by the signal peptides from secretory proteins as well as that present in the signal peptide for lysosome and certain membrane proteins. A model for the function of the SRP. At first, this particle is free in the cytosol in a dynamic equilibrium with SRPs bound to ER membrane. The free SRP binds to ribosome with low affinity. Later on, when the translation of signal peptide starts, the affinity increases. With the emergence of signal peptide from the ribosome, the SRP binds to it and synthesis is arrested. This situation continues until ribosome attaches to the surface of the ER. The attachment occurs by the means of ribosomes and SRP receptors present in the ER membrane. When the final assembly of ribosome with the ER membrane is produced, the block of protein synthesis is released and the polypeptide chain resumes its elongation and is translocated through a pore-like structure. It was suggested that interaction ribosome signal recognition particle receptor may be transient and mainly involved in the targeting of arrested ribosome to a specific site on the ER membrane. Later on, the interaction might be replaced by a direct interaction with ribofourin 1 and 2 which are stoichiometric amounts with respect to the membrane-bound ribosome. In conclusion, the main role of the SRP is that of stopping the synthesis of protein after the signal peptide has been exposed from the ribosome. By this mechanism, the ribosomes making secretory lysosomal and membrane proteins such as albumin, acid phosphatase, and glycophorin are recognized and have time to find the site of attachment to the membrane necessary for the translocation and further processing of the polypeptide in the lumen of the ER. The SRP appears to be indispensable component of machinery for protein synthesis that function as an adapter between cytoplasmic translation of protein and its translocation across the membrane, Walter and Blobel. Protein secretion in bacteria uses a signal peptide. Proteins that are secreted by bacteria follow a pattern similar to that of eukaryotic cells. Although bacteria lack an ER, they are able to secrete proteins that attach to periplasmatic space and cell wall or that diffuse in the environment. For example, bacteria that are resistant to penicillin secrete the enzyme penicillin, and Corynebacterium diphtheria exports diphtheria toxin. The synthesis of these and other proteins occurs on ribosome attached to the plasma membrane. Difference between protein synthesis on free or membrane-bound polysomes in bacteria by the use of different labels that do not penetrate into the bacterium. It is possible to demonstrate that the proteins that are secreted become labeled while the ribosome are still attached to the membrane. On the other hand, the proteins produced on free ribosomes remain unlabeled. Bacteria offer the possibility of studying mutants in which changes in the amino acid sequence of the peptides are produced. The substitution of a simple hydrophobic residue may be enough to abolish the translocation of the protein. As expected, transport was most affected when hydrophobic amino acid in the signal peptide were subtunneled by the charged amino acid. The molecular mechanism involved in the synthesis of proteins will be studied in chapters on molecular biology. Although this process occurs in cytosol, the polypeptide chain of exportable proteins grow into a groove or tunnel in the 60S ribosomal subunit and penetrates through a channel into the lumen of the ER. The signal hypothesis tries to explain the mechanism by which the mRNA is able to recognize free or bound ribosome. It is postulated that the mRNA for secretory proteins contains a set of special signal codon localized after the initiation codon AUG. Once the ribosome recognizes the signal, the ribosome becomes attached to the membrane and the polypeptide penetrates. It is also postulated that the luminal surface there is a signal peptidase that removes signal peptides. Thus, the mRNA produces a pre-protein of larger molecular weight than the final protein. The signal peptide has been 15 and 30 amino acids, which are generally hydrophobic. Such a signal peptide probably established the initial association of the ribosome with the membrane, but some protein factors are involved. An SRP complex bound to the nasal signal peptides and stop the translation until it reaches the ER membrane. It has been postulated that a docking protein removes the SRP block, allowing for the translocation of the polypeptide. A similar mechanism of protein secretion by a signal peptide is found in bacteria that secrete penicillase or diphtheria toxin to the medium.